good old mosses. It won't take you very long until you can tell a moss from a liverwort. And once we go through this, I think that at least you'll be able to tell the major groups apart pretty well. If you see this kind of a leafy plant that has leaves that are arranged around the stem, you're almost sure to be dealing with a moss. There are leafy liverworts, which we'll look at after the break, but their leaves are usually arranged in a flat kind of a way, a dorsiventral way, so they're easy to tell. And there we can see the sporophytes here. They have characteristics that are really different than liverworts. Remember, the mosses have this kind of broomstick or broom straw sort of a seta, much more perennial kind of a thing. And then this strange structure that's up on top of the sporophyte we're going to talk about, that's a dead giveaway for a moss. This is an extreme example, but that's called the Calyptra. That's a structure which is on top of the sporophyte, on top of the capsule. And this particular genus, you can always tell, even with a hand lens, because it has these enormous calyptrae, which are like a candle snuffer that completely covers the candle. So Calyptra refers to that its Calyptra encloses the capsule. So this group, for one, if it's got sporophytes, you can tell it right off with a hand lens just because of the Calyptra. So these parts that I'm talking about, this is just a little tiny fragment of showing you what some of the diversity is, that mosses are really different from each other. You've got all kinds of different leaf arrangements and different leaf stances, and the sporophytes have a short seta or a long seta. Here is another group which has, you can see the Calyptrae on there, which has a very different kind of Calyptra. Those have hairs on it. That's the genus Trichome, which means straight hairs. So that genus is named because of the hairs that are on the Calyptra. We see these more rodulate forms that look a little bit like flowers. We see flat ones. So many different, there's somebody in the waiting room, Stacy or Amy. So what are the characteristics of mosses in general, exceptions to everything? But these are all leafy. None of them are thalloid. They are usually erect, or if they're laying on their side, they at least have the leaves coming out around the whole stem. They can be, and I'm going to show you pictures of this later, they can be upright in tufts called acrocarpus, or they can be creeping across the substrate with a lot of branches and the sporophytes coming from the side, which is called pleurocarpus. That's a early key lead that you come to in keys. As we've looked at before lunch, the leaves are 1 cell thick, usually. There are some that are characteristically 2 cells thick and some more. Great key characters if you get a moss like that. Mostly they're spirally arranged around the stem, although as you can see in the upper right here in the genus Fissidens, Fissidens, Fissidens has actually a flattened morphology where the leaves are on just 2 sides of the stem, but then you can tell this genus at a glance. The midrib, and I'll show you what that is in a minute, moss leaves often have a central strand of conducting tissue, whereas liverworts never do. The leaves are either entire or they're serrated, but they never have a lobe in them in the way a liverwort does. Liverworts, as we'll see later, are fundamentally lobed, have 2 lobes, whereas moss leaves have just one main body. So they're never like bifurcate, but they are often serrate. Oil bodies are not present in the mosses, but we haven't seen them yet. They're in the liverworts, and we'll see those in a little while. We have talked about the seta, the stalk for that the capsule is born on. It is more long-lasting and grows over an extended period of time. So if we look at some of these traits now, let's first go over these parts again and see the Calyptra you can tell is really different here. In the genus Calyptra, it enclosed this whole capsule. More commonly, it's pitched up on top of the capsule and it doesn't enclose it all the way around. This has a split up it like a bishop's mitre. So this would be mitrate Calyptra, named after the bishop's mitre, like those hats that have a split up one side. And the origin of that developmentally is kind of interesting. If we're doing a botany test for people, we ask what ploidy that is, because the whole sporophyte is diploid, the seta and the capsule. But this little bit, the Calyptra, is mother gametophyte tissue. So it's haploid and it's carried aloft as the sporophyte grows. And believe it or not, is actually the remnant of the Archegonium. We looked at Archegonia before lunch, the structure where the egg cell is born. After fertilization in mosses, particularly, the Archegonium develops actively itself, and it becomes this protective sheathing structure around the sporophyte, and so is elevated up with it. The type of Calyptra is a key character, whether it's split down one side, like here, or whether it's whether it's entire all the way around it, like here. So there's one of your key characters right there. When you can tell the major groups apart with just a dissecting scope, but when you really want to get down to the nitty gritty and tell the species apart in a group that is diverse, it's really handy to have the microscope. These slides here these are microscopic views that are prepared by taking the moss plant and hydrating it, putting it in a little beaker of water, and then scraping the leaves off of the stem. Here's a whole stem here where the leaves are still attached. But if you were to cut those off with a scalpel, then they would be detached like this and a lot easier to see. We sometimes like to look at the whole mount of the stem as well, because there are characteristics having to do with how the leaves are inserted and how the leaves are standing relative to the stem that are useful to see without dissecting it. So what we usually do is plop a couple of intact stems in a little drop of water in a microscope slide. We dissect off some leaves, put a cover glass on it, and then look at it under a compound scope. Because the cellular characteristics are really diagnostic, this particular one, you can see it's not magnified quite enough to show it in detail, but there's something going on at the margin of this one, and it turns out it has different cells along the margin. So that's what we would call a marginal border in a key. This one happens to be serrate. Many mosses have the same kind of cell all the way out to the edge, so they don't have a leaf border. Here's that midrib I was talking about. Many mosses have a single midrib that just goes up the middle, but as an important key character indicating family level differences, some mosses have a double one. So here's this double costa here, double midrib. And some don't even have one at all. Here's the bottom of the leaf of a moss that has no midrib. So there's no differentiated conducting cells in the middle. But this is in here to show you another set of key characters. The angle where the leaf is going to attach to the stem is often has differentiated cells. So right down here, this is where the leaf was attached to the stem. This is a particular characteristic where there's inflated balloon-like cells that are down in these lower corners. Those are called alar cells, A-L-A-R. And a key character, a key lead would say alar cells differentiated or not. And so you'd need a nice clean dissection of the stem so that you can see all the way down to what do those cells look like right where the leaf is attaching. Just like the term that's used in trachophytes, some leaves can be decurrent, which means that their attachment kind of trails down the stem. So that those are leaf stance kind of characters. Sometimes the leaves are imbricate, the same term that you use in Trichophytes, meaning that the leaves are closely pressed to each other, whereas sometimes the leaves are spreading like they are here. This would be spreading. And in some mosses like Centrichia, the leaf is actually curled back away from the stem. So that would be recurved. It's squaros if they're sticking straight out, and squaros recurved if they're actually curved back. Those kind of characters you can see right with your dissecting scope. You can see what the leaf stance is. So the first thing we're doing when we're looking at the moss would be, if we don't have a compound scope, we're looking for the leaf stance. We're looking for the differentiated marginal border, what the midrib situation is there one or two or none? And then if we can zoom in a little bit to the cellular level, then we'll see whether the alar cells are differentiated, or in some taxa, the terminal-most cells are really differentiated. So the key might ask whether there's differentiated alar cells or differentiated cells at the apex, or differentiated cells right along the margin, as you see here. And then when we look up close, we see leaf cell shape differences, which turn out to be pretty much family level characters. So they're really deep, evolutionarily conservative characters. This moss that's pictured here has linear cells that are long and skinny. So they're more than 20 times longer than they are wide. But other mosses, including this one here, even though you can barely see it, but I'll show you some later, have isodiametric cells where they're basically squares or possibly circles. Or one whole group of mosses, the Bryophytes, has hexagons. So its leaf cells are always a hexagon shape. So you know you're dealing with that group of mosses almost instantly just by the cell shape that they have. So the cell shape is also important. And then we were talking about papillae when we were talking about water relationships, which would be bumps on a cell. A very important and fairly high level key character would be whether the leaf cell surface is smooth or whether it has bumps on it, like those antler-like projections that Centrichia had. This one that we're looking at close here has smooth cells. It does not have any projections sticking out. The projections can be of a couple of different kinds. Sometimes the whole cell surface bulges up, which is called a mamilla. Sometimes you have little separate bumps like in the Centrichia, which are called papillae. And then sometimes the end of the cell projects out of the plane of the leaf. A bit like if you had a train derailment and the cars that were behind the cars in front kind of pushed up to the side of them, you get that kind of a train wreck papillae on some of these where just the end of the cell is sticking up above the plane. Not sure what the functional significance of that is, but when you see that, it's just characteristic of certain genera and families. So there are really many, many characters all in the gametophyte. And then when we get into the sporophyte generation, I'm going to show you a close-up of some of these characters in a minute. But we have our capsule, and we have this beautiful structure that's at the end of the capsule of most mosses, which is called the peristome, which means around the mouth. And that has really important taxonomic, very high-level taxonomic characters in it. It's a bit like the flower is in angiosperm. Many of our higher-level groupings of mosses are because of peristome characters. Or Calyptra characters. Here is Politicum, which translates to Polytricum, translates to many hairs. And it's one of these ones that has a hairy Calyptra. So its common name is the haircap mosses. If you're familiar at all with mosses, you would just instantly look at that and go, oh, that's got to be Politicum. So even with your hand lens, even with a dissecting scope, you can tell Politicum. To really tell the species of Politicum, for example, you would probably need a compound scope. So if you're not working with a compound scope at the moment, you wouldn't necessarily be able to get all the way down to the species level, but you could certainly recognize most genera, even just with hand lens probably. So those are the kind of characters that are varying. And there are all kinds of life cycle differences. We were talking about the ephemeral ones. This little guy here, Fiscal Mitrium, is one of the ephemeral ones that you would get to see in the spring in a normal year in California. I hope it's out there. That just has a tiny little gametophyte. This whole thing is really teensy. And then it has a tiny little sporophyte sticking out. And you can see its Calyptra there. But it has almost a bulbous look to it. This family and this family, this is Funariaceae. They can be told from their leaf cells, they have rectangles for leaf cells, long rectangles. So they can be told from Bryum or Bryophytes just because of their leaf cell shape. Here's a close-up of a Politicum. It has 4 angled capsules, which is another trick to recognizing Politicum. Instead of having round capsules, it has 4 sides. It would be a square in cross-section. And you see its little Calyptra perched right on top of it with the mini hairs, giving the name Politicum. And this shows what's underneath the Calyptra. If you take the Calyptra off, you have a lid on the sporophyte, which is called the operculum. So the operculum is inside the Calyptra, and the peristome is inside that. So to get to the peristome, the capsule has to open up, and this operculum has to come off, and then the peristome will be inside of it. Here's different growth forms, different mosses. This is a really common moss that I'll bet you have in your garden. It also is a fire-following moss. In nature, it's the first one or one of the main ones that comes in after a fire, even in the first year. We're not sure if that's coming back entirely from a spore bank or also from spores that blow in, but it has an asymmetric capsule. That's why I put this in to remind myself to tell you that not all of the capsules are symmetrical like a Politicum is. These are bent over. They have a definite curve to the capsule itself. And their Calyptra is sticking out here like a little heron's beak. This looks like some sort of a bird sitting on top of the stalk with its beak sticking out. Funaria, absolutely easy to recognize. This is when, once you learn this, you can amaze your friends and associates by just picking it up from somewhere and just looking at it with your hand lens. It has very bulbous gametophytes that look a little bit like an onion bulb with a bunch of leaves up, imbricate, and oppressed on each other. And this one is an annual also. So it can live from year to year, but it does really well as an annual. So here's this distinction between Acrocarpus and Pleurocarpus, which is always at the beginning or near the beginning of a moss key and is confusing to people. And if we break down the roots of these a little bit, it's easier to remember which is which. And the distinction between these two is not a phylogenetic one so much as it is a growth form characteristic. So it's a little bit like shrubs and trees and herbs. Those don't characterize big taxonomic groups, but they're also really useful for a quick characterization of a plant. So in general, mosses either grow in a tuft, which is a bunch of upright stems that are growing next to each other, making a little button or a tuft, or else they're growing laterally and making a mat. They're growing, they look a little bit like a fern. Sometimes those are called the fern mosses. So they're growing laterally and they cover the substrate in a different way. Maybe we can talk about that in the discussion about what would be the ecological consequences of being one or the other of these. And the ones that grow in tufts, upright, they bear their sporophyte at the top of the stem, which comes right out at you. This is one of the tortulas related to Centrichia. You see the sporophytes here are nested right down in the top of a stem. And you can see all their characteristic parts. You can see they have their Calyptra still on. This is actually, that's kind of cool. That shows you what the Calyptra is doing when the stem is still growing. So the capsule isn't fully mature in this one yet. It's completely covered like an Encalypta when it's immature. And then as the capsule develops and expands, it splits the Calyptra open and gives you this. There have been several recent studies on what the role of the Calyptra is in its physical and hormonal control of the development of the capsule. So it's like maternal care that's going right along in development as the new generation elevates up away from the mom, which is down here. So these are born on top of the stem, where with a pleurocarp, you see the sporophytes are coming out on little short lateral branches. So they're not at the very top of the stem, which we can't even see. It's off the bottom here of the picture. So let's break down the roots. So acrocarpus, carpus is a general word in Greek for fruit, which by fake homology or analogy, mosses capsules are often called fruit. So you can see, in a book where it'll say, oh, this is in fruit. And of course, we don't mean fruit like angiosperms. We just mean it has sporophytes, it's fruiting. But acrocarpus, acro means on top of. It's the same root of a word that you would use in acrobat, where two people are on top of each other. So acrocarpus is easy to remember. It means on top of fruit. So it means these quote, quote, fruit are coming right out of the top of the stem. Whereas pleural means side. Like when you have pleurisy, you have a side ache. So pleural means the side. So this is side fruit. That means the fruit are coming out at the sides. That's an easy way. An easy mnemonic is in botany is often to learn what the Greek roots are of the words. And if they're descriptive, it can definitely help you remember them. This Alcia, I don't know how many of you seen, I'll bet a lot of you have seen this one, Tortula muralis. Mural means wall, like when you have a mural that somebody paints on a wall. So this, from the Greek root of its species name, you think, wall. Well, it likes to grow on walls. So if you have an older brick wall that has concrete exposed, or on the side of rocks, you can almost always find this. This really likes, for some reason, it likes to grow on walls. The Duke Chapel in North Carolina had a lot of this growing on the walls. We used to go get it to show people. So that one you probably have seen, actually, and you might even have some around your neighborhood. Alcia Californica is one of these unusual endemics, that's endemic at the genus level to not just California, but to the West Coast of the US. So it is also in Washington and Oregon, and it's really close to the coast. So you don't find it in the Sierra Nevada, it's in the coast ranges, and usually in not the inner coast ranges, it's the mountain range that's the closest to the ocean. When you go to the second ridge, that's, one more removed from the ocean, you don't find it. So it seems to be really maritime loving, maybe humidity. But you can find this in all over the Berkeley Hills, for example. It's very common pleurocarp on the trees there. And it illustrates one thing that I did want to show you that's another key character. Do you notice how the bottom of the seta is sheathed by these really long leaves? If this was an angiosperm, you'd call them bracts, and you could call them bracts here too. So in both angiosperms and in bryophytes, a bract is a modified leaf that's often doing a protective function kind of thing. So these are called, paracaetial leaves. And a key character could be, and one of the characters for Alcia would be that it has differentiated paracaetial leaves. Most mosses, like this tortula here, the leaves that are right around the sporophyte are the same as the regular leaves. They're not differentiated in any way. So they're not differentiated paracaetial leaves in most mosses, actually. But in some, there are highly modified leaves around the sporophyte that have a completely different structure and leaf cell structure and shape and everything. So if you see a pleurocarp in the ridges close to the coast that has these long sheathing paracaetial leaves, you got Alcia. It's relative, it's a pretty deep relationship, but its closest relative is that Dendroalcia that we were talking about earlier, which is quite similar looking to it in many ways. But I don't know if I have a picture of that in here or not, but most people do know that one. It's the little fir tree moss. They both have plumose kind of growth form, which is like a plume on a hat. And they kind of replace each other. Dendroalcia likes it a little bit warmer. So when you go in from the coast and into the Sierra, you'll find Dendroalcia, but not Alcia. Here is another feature that you look for. The ones that we were looking at here and here, the capsule is straight out from the seta. You'd call this erect. This one isn't really erect because it's growing from the side of the stem, but at least the capsule is just a straight extension of the seta. Then you notice how here the capsule has a bend in it, so the capsule is like basically upside down as compared to the seta. This is quite characteristic of certain groups. Just looking at this, without even looking at the gametophytes, we're guessing this is something in the Bryaceae group. So this has nodding capsules that are hanging upside down. The Calyptra's gone, but the operculum is still there. Once that operculum goes, you would have the peristome exposed, and we'll look at some of those later. Here's another one that has these nodding capsules. These have the operculum off, so you can see the little teeth right there. And then if we zoomed into any of these leaves that are in the Bryaceae group, we would see they're the ones that have the hexagons on their leaf cells. So the combination of those two and just the look of them, Bryum is one of these groups like sedges that is very hard to tell the species apart, but the general group is really easy to tell. It's quite easy to tell that you're at least dealing with a Bryum or one of the segregates. Rhodula Bryum is a segregate of that in general. Okay, so now let's talk about the peristome. So that's, once the operculum's out of the way, like it is here, you have these fabulous little structures that occur around the mouth of the capsule, and they only occur in mosses. They're not in liverworts, not in hornworts. So they're actually quite easy to tell. You know you have a moss if you see a peristome. The one on the right here is a regular microscopic view, and then the rest of them are scanning electron microscope views of what the peristome looks like. And of course, I have to feature Centrichia. Trichome means hair, and it has that same meaning in the vascular plants as well. If you have a trichome on a leaf or something. So trichome just means thread or hair in Greek, and syn means together, like synergy. So Centrichia kind of means, threads together or hairs together. And that refers to this amazing peristome, which has these twisty filaments that kind of bind themselves together. At the bottom of the peristome starts way down here. That's the top of the capsule. So the bottom of the peristome is actually, you could say, woven together like a basket. This is called a basal membrane. And then the segments become free partway up, but they twist around themselves. So this peristome is just absolutely unique to Centrichia. That Physidens that we saw in a few slides ago, when you zoom in, they're just beautiful. I love the look of all these guys. Now, one big distinction is whether you have a single row of peristome teeth or a double row of peristome teeth. I'm going to show you phylogeny in a minute, but that's like a really big character that has a big division within mosses. Centrichia, Physidens, and Grimmia, which is here, are all single peristome mosses, and they're members of a huge clade of single peristome mosses. Whereas the ones that are nodding, like those Bryums we looked at here, When we look at them up close, we see they have 2 rows of peristome teeth. And if I point them out to you, you can see they're really different from each other. There's this outside row right here that's kind of curved in. That's called the exostome, which means the outside peristome. And then there are these much finer filamentous versions in here that are called the endostome or inside peristome. So many mosses have 2 very distinct rows of peristome teeth, and many others just have a single row. And it really is phylogenetically significant. I don't know how functionally significant it is, but maybe we can talk about that. The nodding capsule ones tend to have this kind of peristome, whereas more upright ones tend to have the single peristome. Maybe we can talk about that in discussion, what the role of this might be. But since we're talking about taxonomy, I want to point out that in this case, the endostome is alternate with the exostome. So you have an exostome tooth, then an endostome, then an exostome. So you call this an alternate peristome, alternate double peristome. In some groups like the Phanereaceae that we were looking at a minute ago, the endostome is exactly lined up with the exostome. So that would be an opposite peristome arrangement. So we even use formulae for peristomes, a little bit like the floral formulae you might have learned in botany to tell angiosperm families apart. So just like some angiosperm families have the calyx and the corolla alternating, and some have them directly opposite each other, it's the same kind of thing. So again, we could maybe talk about what the functional significance of all this is. I'll give you a hint that these teeth are actually hygroscopic, which means they open and close with humidity. So they're amazing. This is what the dry arrangement looks like. In Grimmia, they're all the way bent back. So the peristome is so open that it's kind of bent back. But when conditions are wet, these teeth are bent inside and cover the opening of the capsule. There's weird stuff going on with the double peristome mosses. The endostome is not hygroscopic, but the exostome is. So the exostome usually opens when it's dry and closes when it's wet. We could talk about the significance of that. But it's beautiful. And for scale, you can see the spores here. We don't know all the functions of the peristome, but we think it has to do with spore dispersal and regulating spore dispersal. But whatever the functional value is, it has great taxonomic value. So the keys will ask you about that. Okay, so I wanted to look at one more website in detail that we mentioned before lunch, but just to walk you through it since I didn't have it handy. We host this on the website at UC Jepson, but it has been adopted by the Bryophyte chapter. It's not as slick. It's set up like the Jepson e-flora for vascular plants, but it's not quite as slick because we don't have any staff. So it's all volunteer effort. So if there are people out there who are interested and want to contribute, we're always looking for volunteers to help us with this. Someday we'll be able to afford to have actual staff to run it. But what we basically did was we started with the Madronio volumes that you may know of that were published in the 2000s. There was just a catalog of the mosses of California, and then there was also a set of keys by Norris and Shivak. We took the content from that and put it online, and then we've been working on it as we get time to add new taxa and to fix it up and so forth. You can buy these if you want to still from California Botanical Society, but the content of them all is put in here online for free. If you guys have never used the Keybase program for either the Jepson e-flora or this, I definitely recommend you check it out, but I'm not going to go through that. What that lets you do is key out taxa that are only from a certain area. It filters There are filtered keys that filter out taxa that are not expected in an area. It does make it a lot easier to keep things out. But I wanted to show you kind of what the key to keys would look like. This would be if you're like in the Jepson eFloor or the Jepson manual, if you have no idea what something is, you have to start in the biggest key. If you already know what genus it's in, then you can go right to a genus and you can pick the key. So once you become more expert, you don't have to go through it. So Brian, we were just talking about a minute ago, that's one that has these nodding capsules and hexagons. And, you know, I'm not going to go through it with you, but these key characters are a lot harder. You need a microscope for these. It's asking you for the leaf cell, you know, shape and size, and where there's leaf borders, and whether the costa is another word for midrib, whether that's sticking out of the leaf or it's contained within the leaf, leaf cell width, leaf size, capsule characters. You know, Bryum, I shouldn't have shown you this one first because Bryum is one of the worst, the most difficult groups. It's definitely the carex of the mosses. There are many other groups that are quite easy. There's good old Alcia that we were looking at. And Alcia is easy because there's no other Alcia. There's just, it's a genus level endemic as both it and the Dendrocea are. But so if you go to your basic key, the key to keys, then you're going to see a lot of the characters that we were talking about. Here's E costate, that means no midrib. So that's one thing we were talking about really early on. Some leaves have hyaline on, so they have a hair point sticking out like Centrichia, and many don't. You have, you know, many other of the distinctions we were looking at, whether the leaf cells are papillose or smooth, and so forth. So you could handle, here's double costa and no costa, and somewhere in here is going to be pleurocarp, acrocarp pretty soon. So you could probably work your way through this with a little glossary. And there are some decent glossaries for these characters if you want to, you know, want to check out if you don't recognize what they are. But it's not all that difficult. Then this would get you to a genus key, and then you go to the genus key. So that's just a little background to this. And then once you get to a species page, again, we need more work on these, but we try to have a picture of the moss and a description of the moss if we have it. And I believe that we do have it for these. We have a little, you know, eFloor type map and a description if we have it. Dan Norris wrote many of these. So there are not descriptions for all of them because Norris unfortunately died before he finished all of the descriptions. But we're working on this. We don't have any illustrations in our own database. So I definitely suggest you explore this. This is unfortunately just for mosses. We don't have one of these for liverworts yet. But if somebody had the if the spirit moved you to do that, then we'd certainly support it and try to help. If you wanted to have a liverwort eFloor, that would be great too. Okay, so to sort of summarize where we are with the mosses are this clearly defined monophyletic group. And I think before I delve into here, I want to take a short break, and then I'll come right back and I'll talk about how all these different mosses relate to each other and take some questions and things.